Hello folks, this lesson is going to be on a single trait or monohybrid crosses. These are the most simple of the crosses where we're only taking a look at, well, one single trait at a time. And for this example as well, for this particular gene, there is only two different possible alleles, and it is a straightforward dominant recessive relationship. So in reality, it doesn't really get much simpler than this example here. A couple of points that I do have at the top. So first of all, just a reminder that when we're talking about adult organisms, the adult organisms, they are going to have two copies of each gene, one on each of the chromosomes, one from the father, one from the mother. So of course that is referred to as diploid or 2N. When we're talking about the gametes, the sex cells, the sperm and the eggs that are formed in the testes and the ovaries, they are formed through the process of meiosis, which is reduction division, which means we're taking the diploid cell and we're converting it into haploid cells that have only one copy of each chromosome. So to determine the possible genotypes and phenotypes, what we do use is we set up a Punnett square to kind of organize things. And I have a couple of Punnett squares at the bottom here that eventually we will fill in. So in this example here, we're taking a look at the wool color in sheep. And for this one here, again, there are only two different possibilities, two different possible alleles, and two different phenotypes as well, and that is the black and the white. But let's go ahead and read through this example very, very carefully, and I'll show you how I would go ahead and solve a problem like this. So what do we have here? Well, we have a black wool sheep. Curves only, and this is kind of important, the only way that you can get the black wool sheep is when the sheep is homozygous recessive. Homozygous recessive. So you need to understand the terminology. Remember that homozygous means that the letters, they're going to be the same. We use capital and lowercase letters for dominant recessive relationships. So just up here, I'll just write that, well, what we could use is a capital A. That would represent the dominant allele. Lowercase a would represent the recessive allele. So if we're talking about homozygous, again, in an adult plant, or an adult organism, in this case the sheep, there need to be two copies that are the same. So that could be either two big A's or two little a's. So black wool sheep, only when we get homozygous recessive, so they would have to be two lowercase letters, but what letters are we actually going to use for this example? So let's take a look at the rest of the question. It says calculate the genotypic. So again, you need to understand the language. What are we talking about when we say genotype? And the phenotypic, what are we talking about when we say phenotype? And for genetics, we do talk about ratios. So a ratio could be something like, for example, a 6:1. That would be a ratio. Again, more of these sort of terminology that we use. F1 are the offspring or the children, and F2 are the great-grandchildren. So I'm asking for the next two generations after the parental generation when you're filling in the eventual Punnett squares here and making your prediction. So calculate the genotypic and phenotypic ratios for the F1 and F2 generation when you're crossing. So what are you actually crossing in terms of the sheep? Well, we have a black male sheep and we have a homozygous, again, that's going to be kind of important, white female sheep. That is the cross that we are doing. So the very first thing that you need to do is you need to determine what letters are you actually going to go ahead and use for this cross. So let me just erase some of this mess that we have here, make it a little bit cleaner so I can add a few more things to this example here as I'm kind of modeling this with you and working our way through this problem. So the letters that we're going to be using. So remember that we use a capital letter for the dominant allele. We use a lowercase letter for the recessive allele. So we know that the black is going to be recessive because we're well told the black is recessive. Just trying to get my pen back here. There we go. So the black is going to be the recessive. because we're told that we can only get it when we have homozygous recessive, which means our white, in the case of the female here, is then going to be the dominant allele. So the format that we do use, and yes, once again, this is kind of important, this is what we do in genetics, is we choose the first letter of the dominant trait, we capitalize it, and that is the letter that we do use to represent this particular trait. So we would then use a capital W. So capital W is the letter that we are going to use to represent the dominant white allele. What about the black one? Well, this one here is recessive, so it's going to be lowercase, but it's not the B that we're going to use. Again, the format that we do use in genetics is to use the same letter, which is a W, only now it's going to be lowercase. So often I'm asked, well, why is it done this way? Do we have to do it this way? And the answer is, well, yes, this is the way that we do it in genetics. So this is the proper way to assign the letters that you are going to use for the dominant and the recessive alleles. So step one, assigning the letters, and make it very, very clear what letter you are using for which trait, so that you will use to represent each one of the traits. What are the traits that we have? Well, the traits are the dominant one, which is white, and what we're going to use is a capital W. And again, you need to tell me this, and you need to make it very, very clear. And the other one, which is the black allele, the black version of this gene, is going to be the lowercase W. So now we can go on and we can talk about the actual genotypes of the parents. So once again, parents, they are diploid, which means they're going to have two letters. So kind of like what we have up here for the A's, only now we're using W's. So what are the different possibilities? Well, we can have big, big, we can have big, little, and we can have little, little. So because the capital W, the white, is dominant, both cases here, this one here is what we call homozygous dominant, this one is homozygous recessive, and in both cases then, because there is a capital W, capital overshadows the recessive lowercase allele, so in both cases here, what we would have is a sheep that looks white in color. So different genotypes, but they look exactly the same. The only way that we can possibly get the black is if we do have the homozygous recessive, and in this case, that's the two lowercase w's that we would have. So the genotypes of the parents, what would then be the genotypes of the parents? So for our male, this is the symbol that we do use for the male. So the male is the one that has the black wool. Trying to write here as carefully as I can with this pen. Let me erase that, try and do it a little bit better. Okay, so that's our black wooled male. And big X just means that this is what we're crossing it with. And this is going to be our female, of course. And this is our white wool female. So genotypes. These are the phenotypes. What are the actual genotypes? Well, the only way, once again, to get black is if we have two lowercase letters. So this is really what we're crossing in terms of the genotype. It's two lowercase times, and the case of the white female. So is it big, big, or is it big, little? Well, the question actually tells you that. And it tells you that the female is homozygous, which means the letters have to be the same. So the only possibility then is going to be our big, big, two capital W's in the case of the genotype for the female. So now the purpose of the Punnett square is to make your prediction for the next generation. So the Punnett square on the left is the F1 generation. We have these two parents. This is the parental generation, and we just abbreviate that with a capital P. And we're going to cross these two parents. The parents, of course, are diploid. Once again, they have two copies, two W's, two copies of each gene. They might be the same allele, they might be different, but they are diploid. When we talk about the gametes, though, they're only going to have one copy. So what I'm going to do in this Punnett square is across the top in these two spaces here, I'm going to put the gamete genotypes from the male. It doesn't matter if you put the male along the top or the male along the side. In this case here, I'm going to put the male along the top. So when the gametes are formed, the gametes do not get both capital W's, they will only get one. So half of them will get this one, the other half will get this one here. Same thing with the female, they're not going to get both. Half are going to get, well, this allele, and half are going to get this one here. So if I now go ahead and just write in right at the top of the Punnett square and the side, what we're going to get for letters, this is the way that you would then write it in. So again, I'm putting the male at the top. So we would have a lowercase w. In fact, it's going to be a lowercase w in every single one of the sperm because that's all the male has, are the lowercase w's. Similarly with the female only has the capital W's. So in this case, every single one of the eggs is also going to have the the capital W. So now what goes in the square, this is important, this is your prediction. What you're saying is this is what I think is going to happen based on probability in terms of the genotypes and phenotypes in the next generation. The way that we fill in the square is just the intersection. So this lowercase w is going to go down, this capital W is going to go over. You always write the capital letter first. So we would have capital W and a lowercase w. What about the next square? Well, lowercase w comes down, capital W goes over, and again, this one is also going to be heterozygous. And in fact, all of them in this first Punnett square that I'm drawing here are going to be heterozygous. So now we do need to write the genotypic and the phenotypic ratios from this particular Punnett square. So the way to write a ratio for the genotypes is really to write what the different genotypes are. And what we do is we write the homozygous dominant first of all. So we would write this as big, 2, the big little, 2, the little. So when we take a look at this, well, clearly we can see, in fact, we didn't even really need all of these ones. We only needed one square in this Punnett square. And what we can see is that it's 0, 2, 1, 2, 0. If they're all exactly the same, you don't necessarily have to do this and write it as a ratio. You can just tell me if they are all the same. You can just say all are, well, in this case, heterozygous. For the phenotypic ratio, what are the phenotypes? And you should write the dominant one first, the white to black. So if we take a look at this one, all of them have a capital W. So that means that we would have a 4 to 0 ratio. You always reduce all of your ratios, by the way. So if you do end up with, let's say, a 4 to 2 ratio, that's reduced to a 2 to 1 ratio. So this one here we could just say is a 1 to 0 ratio. But once again, if they are all exactly the same, you don't need to tell me in the form of a ratio. You can just say that they are all, in this case, going to be, and I'm getting kind of messy as I squeeze it in at the bottom here, they would be all white. That's not all that terribly interesting, but now let's go on to the F2 generation. So now the offspring in the F1 generation, they become the parents in the F2 generation. Let's get rid of some of this over here so I have a little bit more space. So I'll write it off to the right-hand side here, but what that means that we're crossing is we're crossing offspring that are heterozygous. So these are the F1 generation, they are the offspring, but now they become the parents in the next generation. So I'm going to call these P2. So just like we did before, the gametes are only going to get one of the two letters. So now it's a little bit different because half of the gametes will have a capital W, half of the sperm will have a capital W from the males, and half of them will have a lowercase w. This would then be the female over here. So in the eggs as well, half of them would end up with a capital and half of them with a lowercase w. So we'll put our capital in here, lowercase here, fill in our Punnett square just like we did with the F1 generation. So this one is then big, big. This one here, big, little. Again, capital letter is what is written first. So big, little, little, little. So yeah, this one's a little bit more interesting for the F2 generation in terms of the ratios. And this one here, we do need to actually then write as a ratio. So again, the format, just as I showed you before, is to do the big, big, homozygous dominant first, heterozygous, big, little second, and homozygous recessive third. So if we take a look at this, we have 1 out of the 4 in the Punnett square. That's going to be the homozygous dominant. We have 2 of them that are heterozygous, and we have the remaining one, which is going to be the homozygous recessive. So we have a 1 to 2 to 1 ratio. For the phenotypic ratio, once again, write the dominant one first, which is the white colored sheep, and the other one, the other possibility, the only other possibility is the black. So now remember that these ones here, even though they have different genotypes, they're going to look exactly the same because both of them have the dominant capital W, which is for white. So 2 plus 1, we have 3 out of them based on probability that are going to be white, and 1 of them based on probability that is going to be black. And I do want to stress that this is a prediction of what we would get in the next generation. So yes, it is possible that these 2 sheep have 4 different offspring, or sorry, have 4 offspring. It is possible that all of them, in fact, could be homozygous recessive, and all of them could have 2 lowercase w's, and all of them could be black. But based on probability, our prediction would say that this is what we would get in terms of the ratio, and that is a 3 to 1 phenotypic ratio. So this ratio as well, this in fact is a really, really important ratio. So whenever you do have a cross between 2 individuals that are heterozygous, I'm just going to use some different letters here. So in this case, I'll use B's. So if these are the 2 parents that we are crossing, sorry, that should be a B, of course, in the first one that I'm drawing here. Let me go back, fix that up. Whenever you do have a cross, a single trait cross with 2 parents that are both heterozygous, what we are going to end up with is a phenotypic ratio, which is 3 to 1. Always tell me what the 3 is for and what the 1 is for. So up here, the 3 is for white, the 1 is for black. As it turns out, though, the 3 will always be for the dominant trait, and the 1 will always be for the recessive trait.